Everyone, thank you. If we can make a start. Thanks for coming along this morning to the first of this series of seminars. My name's Ian Tote from the James Sutton Institute and the National Potato Innovation Centre, and it's a real pleasure to welcome you all here today. We're going to hear today about late blight, a problem that never quite goes away, and we're going to listen to it from three different perspectives today. Before we do, there's a little bit of housekeeping. The first one, if you wouldn't mind putting your phones on silent. In fact, I'd better do mine. Have you guys done yours? And then we won't be interrupted. Secondly, and I'm not sure why you need to know this, but I've been told to say the toilets are just outside. I think that means try not to do it in here. Try and go outside if you need the toilet. There are no fire alarms planned, so if there is a fire alarm, we have to get outside. And I'm sure, well, we're right next to the door, so that should be straightforward. And anybody needing basis or Noroso points, you can get from Ellis, who stood at the door on the way out. So just see Ellis, and you'll see. If you need cards to do that, there are some cards just outside in the foyer. So without further ado, we've got three talks today. First one's going to be from Dave Cooke from the James Sutton Institute. Dave is a scientist at the James Sutton Institute, the head of pathology. He has been at the Institute for almost 30 years, over 25 years, and he's been working on late blight for all that time. For the last 20 years, he's been working with Fight Against Blight and running Fight Against Blight. And he's also a main component of the Euroblight, which, as many of you will know, he plays a very big part in making sure industry knows of the different late blight strains that are coming through and the issues associated with them. So without further ado, I'll pass you on to Dave. Thanks, Dave.
Thank you, Ian. And thanks for the opportunity. And yes, indeed, for you all coming along today. It's going to be a pleasure. You're going to hear the first of this year's 2025 Euroblight results. And I shall run through this in 10 minutes. It's going to be quite tight, so I will press on. If there's anything that you feel you've missed, then we have a few minutes for questions at the end. But do grab me. I'm here for the next 2 days. So it's not only my own work, of course. There's Alison Lees, James Hutton, and James and Beatrix. James and Beatrix are the ones who really do the work on the lab, the samples that come into the lab. So yeah, I'm based in Dundee, as Ian's introduced me to, and yeah, and I've been working there for actually over 30 years now. It is 1994 I arrived. I'd like to start off straight away by thanking the sponsors for Fight Against Blight. I mean, I think it's a part of shows that the work is important, and there are 18 separate sponsors who put their hands into their pockets and support the work. So that's great. GB Potatoes in particular have managed to attract funds for the AHDB funds, and we've benefited from the funds that have come through GB Potatoes as well. Also acknowledging Euroblight and the Scottish Government. Without the underpinning funding that we get from Scottish Government, this work couldn't happen either. The last thing to remember on this slide is the Scouts. So Fight Against Blight depends on many of you probably who are in the field looking at crops day in, day out, who send us samples on, and that gives us the intelligence we need to help manage blight in a better way. So the potato blight life cycle I'm going to go through very briefly here. You'll recognize, sadly, the main driver, which is the asexual stage where the spores are produced from leaves, go through that cycle within 3 days. And the main take-home here is there are billions of spores can be produced. When blight gets out of control, even low levels of blight are producing 1.5 billion spores per hectare at only 1% crop cover. Every spore is a potential mutant and can overcome management practices, that's host resistance or fungicides. So that's the driver. We know that all blight starts from tubers. Almost all of it starts from tuber blight at the beginning of the year. So, the key messages there are obviously healthy seed, avoiding volunteers, which you'll hear a lot more about as we go through. But there is a sexual cycle as well, where A1 and A2 meet each other and go through a sexual phase, and those oospores can survive in the soil for a long period as well. When it goes wrong, not going to dwell on this, just the horror pictures from 2024 due to extremely bad weather and fungicide resistance. I hope none of you have seen that this year. and where we are with IPM. This is all about IPM, and bearing in mind the UK government's 2025 National Action Plan for Pesticide Use, which is focusing on IPM and a 10% reduction in pesticide load indicators by 2030. So, that's the environment we're all moving on to, is trying to remove our reliance as strongly on pesticides and or fungicides, or crop protection chemicals, depending on the terminology, and move to host resistance. And Dan's going to be covering the host resistance, and Andy's going to be talking about the crop protection chemicals. So we're all about managing primary inoculum, looking at decision support systems, effective use of host resistance, when to use fungicide, which fungicides, and that's why the monitoring is important. So Fight Against Blight, we've taken a remake over this year, new website. I hope you've all enjoyed the cleaner, newer interface. Thanks to my colleagues at Hutton for producing that. And where were we? 2025, yeah, every year's exceptional, isn't it? This year was very unusual, incredibly dry, very low light pressure. And I think you can see here on this, the outputs there, the calendars from the Blight Spy, the one on the left with Boston, Lincolnshire. Really astonishing not to have only one half Hutton criteria between January and the middle of July. That's why primary inoculum levels were phenomenally low this year, and that's why blight didn't appear early in the season, even when the weather was conducive, because the primary inoculum was not there. Compare that to Barmouth and Gwynedd, out in the wet, wild west part of the country. A lot more moisture there and a lot more blight activity as a result. So this is where the outbreaks actually appeared in 2025. You can see the spots on the map. It didn't get across to the east of the country until September. So, a remarkable statistic here is that over half of the outbreaks on this map that we registered in this year were from after September. Cutting to the chase in terms of the results, the genotyping, which is done at Hutton using this same technology as criminal forensics. So we'll fingerprint every single strain that comes in, and then we determine which genotype they belong to, and the coloured blocks on the charts here show those genotypes. And you can see the progression over time, and the genotypes have changed, and that's why we're all here. Managing blight is a continually moving, the goalposts are continually moving, if you like, as the pathogen evolves. So what are the headlights? The headlines, I suppose, for this year? We've processed 300 samples. That is lower than average, but not the lowest. So, and there's a big increase in EU46. But the headline figure of 23% EU46 amongst all the samples we've genotyped does rather hide some of the details on that. It's only been found at 7 sites, as you'll see on the subsequent slides. Another key finding is a lack of something, and that's the lack of EU43, which is resistant to Revus. So that's good news that we have yet to find that genotype in the country. I'm going to talk about the other clones later. The population was dominated by EU36. But I'll go through each of these in a little bit more detail before I move on to fungicide results. So EU46 and EU41, the clones we saw this year, this shows where they were found. EU46, we found it first in 2024 in Wales and in Scotland. So we were looking quite intently this year. It popped up again early in Wales on a dump and spread quite rapidly from North Wales to South Wales through the early part of the year when we had outbreaks earlier in the season. And then late in the season, it spread across into Telford, Wrekin area, and then to Derbyshire, as many of you have perhaps seen at the Eurofins trial site. So it was only found on a total of 7 sites. And only 2 of those were on commercial crops, 1 in a garden. So quite relatively small numbers of areas, but we have to consider the fact that blight is still active. The weather's, although it's getting cold, it's not quite cold enough yet to kill all those volunteers. So bear in mind that we are still receiving samples, and it may well spread further than that. But we'll keep you posted. EU41. It arrived in 2021, hadn't been sampled for 2 years, then it went silent, disappeared, appeared again in 2024, and then in 2025 that's appeared again, and you can see the locations. There was evidence of reduced sensitivity to thiazofamid, not resistance, but just a slight difference at low doses that we were keeping an eye on. We think that this stormamy may well have spread EU46. That's the take home there. The winds were in the right direction to spread it from Wales to England. Distribution of EU36, yeah, it's continued to spread. It's now pretty much colonized across the whole country. It is very aggressive, and we found a lot of this on volunteers in the southeast. I'm going to go through these fairly quickly because we do need to move to the fungicide results. The minor clones there, you can see EU37, the fluazanam resistant strain, still very low frequency. That's good news for fluazam use and diversification. EU13, also very rare. EU6, mostly in Scotland, and the others mainly in Scotland, as in previous years. Just to stress, volunteers, thanks to those who provided these images, potatoes should not be so active in the middle of November still get active growth and blight spreading on these things. So bear that in mind for next year. We've obviously got active infections and inoculum spreading. Host resistance, Dan's going to talk about this. So I won't talk about, we are monitoring host resistance and host resistance breakdowns within Hutton and Euroblight as well, and new strains, but Dan's going to cover that in more detail. So onto the fungicide testing, a few minutes to cover that. These are the 5 products we've tested in 2025 in consultation with industry and with sponsors, providing just a bit of independent knowledge on the contemporary populations. So we've tested those 5 fungicides in a detached leaf test, and of those isolates that we've tested there, you can see EU36, EU41 from last year. It didn't come in until late in the season, so we've tested last year's samples. EU46s we've been testing, and the EU43s, we don't have that in this country, but we've been using some Dutch isolates. Cutting to the chase, so oxythiopyprid, the first product here we're talking about, EU36 and EU41 are sensitive, sorry, keyword, very important difference, sensitive, sensitive to Zorvec, which is good news. The EU46 isolates, we've seen them in the past on the continent, and they've been resistant, and the isolates we've been testing this year have also shown to be resistant to EU46. So that's why we need to keep an eye on this, bearing in mind its distribution. Most of the country is still not colonized by this area, so Zorvec will still be effective. The chart on the right shows you the what true resistance looks like. The one on the left there is showing very, very low levels. Always bear in mind the y-axis, the vertical axis on all these charts. The field rate is 75 ppm, so those rates on the first chart are tiny compared to the ones on the right there where we got the AC50s being up at where it is scored as resistant. Mandipropamid. The key take home here again is all isolates were sensitive, all the ones we tested. And if we look at that chart, you can see there is a slight, there's increase, a slight increase with the 43s and the 46s compared to the 41s and 36s, but that's still well below field rate. So we're not talking resistance there. That's just sort of keeping an eye on that. That's what resistance looks like from the data from 2024. You can see that bar is way higher. You can't even see the other data because it's down at such low levels in terms of ppm. Amisulbron. The reason we tested amisulbron was because I mentioned that thiazolfamid and EU41 showed some changes. We wanted to check that because amisulbron's the same chemical group as thiazolfamid, and there were no differences with the EU41s in this year, so that's good news. Propamacarb, again, these are quick. Good news, no change in sensitivity there. Everything was sensitive. But just bear in mind all these single actives. We shouldn't rely on them. We shouldn't rely on single actives in any stage these days. There's so much risk there. So that's one of the take-home messages there. Fluopicillide, similarly, again, although the bars look like different heights, the rates are very, very low there. So we don't need to worry about any resistance to fluopicillide. So that's good news. Last couple of slides we've been testing, all those results are with using live isolates. We can also test the DNA. So we've been doing that with the Sanger sequencing method where we look for the individual base pair changes, and that shows us whether they're resistant or not based on the correlation. That test is slow. We've been using a new test this year, whiz-bang type thing where you get 26,000 separate PCR reactions set up and you can get a higher throughput. And these results show you they show you what a control isolate that's sensitive would look like, control isolates that are resistant, and then some test isolates you can see with ones that are sensitive and resistant and sensitive. We've done a lot more of that type of testing, James has, and again, cutting down to the chase on this, we've tested 80 isolates, 65 of them have worked, and the only resistance we've noted, as you can see from the OSBPI, OXTP, is the Zorvec active ingredient, is resistance amongst the EU46s. Everything else has been sensitive. So, generally good news, and that's a reflection of the way the industry has been using products, I think, very cautiously. So that's good news. We are still testing these samples as we go through. Conclusions? Don't know how I'm doing for time. Okay, right. So hopefully I've given you a very brief and very rapid snapshot on why tracking the pathogen population is important. All the different clonal lineages have different traits. There are elements in which that's breaking down, and we need to bear that in mind. There is variation within clones as well now that we need to think about. But I've given you a flavour of that. Primary inoculum is still something we need to worry about. You won't get blight if there isn't any primary inoculum, so keep control of that. Oospores risk managed by extending rotations as far as we can, but volunteers break down rotations. So getting rid of volunteers and dumps is always the same message. Cultivar resistance is going to become increasingly important to complement fungicides. Dan's going to cover that. I put this up because blight pressure was low this year. Let's not move into 2026 with any complacency. Don't forget, it's still around, and as you've seen, there's a lot kicking around on volunteers at the moment. Fungicide use, again, positive messages. We're all using, I know it's more expensive, we're using more product perhaps than in previous years, but that is important for managing the resistances. And just a last point there about the National Potato Innovation Centre, just awareness of that organisation as well. Ian's running and will tell you about if you want to know more about the NPIC that's being developed to Hutton. And, oops, I've moved on to Andy's slides. So that's it. Thank you very much for your time. I think we're doing questions at the end, aren't we? So.
Thanks, Dave. I was going to cut Dave off, but until I realised he was going to talk about me at the end, so I'm glad that I didn't. Thanks, Dave. So we'll move straight on to Andy, and if there's time for questions at the end, but remember that we're all around today, so please come and talk to any of us throughout the day. So Andy Cunnigham, a potato technical manager at Syngenta, been there since 2016, and since 21 has been responsible for potato and foliar biostimulants, has a big interest in late blight, is part of Fight Against Blight and a funder of Fight Against Blight, and we're very much looking forward to hearing what you've got to say.
Lovely. You just saved me 2 minutes here by not introducing myself. So, so I'm here to talk to you about how we can combat the build-up of resistance in late blight. We know the Europeans really don't know how to control it, so hopefully we can tell them what to do to make sure that they carry on doing what they are actually doing. So I thought really where we should start is this guy here. He obviously didn't really talk much about late blight, but he did say some inspirational things back in the 40s. I didn't know if you realised that. But there was a speech he said in 1948 where he mentioned about those who fail to learn from the past are doomed to repeat it. And I feel like that is very pertinent to how we can control blight and how we can stop it from evolving. So where I wanted to start really was thinking about what actives we have available to us to control oomycete diseases, but also how resistant they are to Phytophthora infestans. And you can see that of them actives that we have in our current toolbox to control that disease, 4 of them actually are showing no resistance at this moment in time, which is really good. It's a good start. And there's also 2 actives in the form of phosphonates, but also zoxamide, which we don't have any data on. But I would say that gives us a good backbone to start an uncompromised blight program. However, if we look at similar pathogens, so Plasmaphora viticola, which is downy mildew in grapes, we can now see that those actives which haven't got resistant issues in late blight do have it in downy mildew in grapes. And that is a very similar pathogen, which shows us that it has the ability to evolve if we use our fungicide practices poorly, okay? So we know the premise. We know this work that was funded through the HDB back in 2019 through to 21 that ADAS carried out. And they showed, quite rightly, that poor use, overuse of single-site chemistry will inevitably select for resistance if it's in the population to be selected for. But there is a positive slant on that. They also showed that compromised chemistry, this study was using flazinam at the time, which is Sherlan, showed that if you use it in mix with another active from another mode of action and alternating it through the program, you can significantly decrease the level of resistant population in there. So what I wanted to talk you through was really how not to do something. And we know that there is the the EU43 strain that is over in Europe, which is more problematic to the active that I look after, the mandipropamid. So I want to show you how not to do things, but also how we can improve things. So this population of EU43 initially wasn't resistant, and it was just a population of 43 that just bobbed along the bottom. But the Europeans do have they did have an issue with how they produce their programs. They use a lot of single site chemistry in blocks and on their own as well, and would be effective at controlling blight, but it would also increase the pressure of selection on resistant strains. Now, in 2021, carrying on them same fungicide practices, they inevitably selected for a version of 43 that had resistance to CAA fungicides. So 3 samples. Unfortunately, without changing them practices in 2022, that population of 3 samples then exploded. And all of a sudden, you've got a problem, right? The Danes, give them their due, did follow UK practice of mixing and alternating, which then meant that they were able to significantly decrease the population of the EU43. That shows you it can work. Unfortunately, the Dutch didn't. And they unfortunately carried on doing exactly what they had been doing and actually created more issues further still. So let's just think about what conditions the Dutch were dealing with in 2023. You can see that red section. Oh, it is meant to be red. Apologize for that. So let me come down. Can I get this microphone? Is that right? Hello, hello. All right, okay. So if we look at the weather conditions during that period, this is around May, the start of June. This is that red box, and you can see that we've got minimal rainfall. So the perception was there was a low risk of blight. As we know, rainfall doesn't necessarily mean you're going to get blight. It is driven by temperature and also humidity. And if we bring in that 90% level of humidity, which blight loves, you can see that during that red section, although there was no rainfall, there was a high proportion of 90% or more humidity. So we actually had really nice conditions for blight to develop. And you can imagine using single site chemistry, the likes of CAA fungicides would have been clearing out the blight population that was there, but we were selecting for that resistant strain. And then following that, inevitably there was spores of late blight pustules, and then we had that significant amount of rainfall afterwards, which then just allowed that population to explode. So in effect, they did actually self-destruct in 2023. They underestimated the blight conditions, which then allowed that selection of EU43, and inevitably, when you get into a curative population, we don't agree with it, but inevitably they do pull out the most potent chemistry in the toolbox, which is OSBPI chemistry. So not only did they select for the CAA fungicide resistant one, they also selected for a version of 43 that had resistance to OSBPI chemistry. And then coupled with that bad level of rainfall afterwards, that small population just exploded. And then they created a really big headache for themselves, but also across Europe. So as it turned out in 2023, they had a massive epidemic, worst for 30 years apparently. There was not only that emergence of the double armoured 43, there was the emergence of the 46 strain, which as we've heard is now into the UK. And the only positive thing is that because of that in 23, in 24, they did change their advice. And as you can see, the population of the EU43 strain significantly declined in the Netherlands. And I don't think we've had the Euroblight data yet, David, but by all accounts, by carrying on that practice, they've reduced that population in 25 as well. That's what I'm hearing. So with all this talk of genotypes, I thought I'd give you a bit of an explanation, particularly around the 43 and how the 46 differs, because there are some key differences with this genotype. Now, how CAA fungicides like mandipropamid work is that they target the enzyme cellulose synthase that the pathogen uses to build cell walls. And without that enzyme, you obviously can't build a cell wall. It has no integrity and it can't survive. What we know with the 43, and along with a lot of other blight strains, not all of them, is that it's a diploid chromosome. So it has 2 chromosomes, very much like we do. And on them chromosomes, there is the gene that will create that enzyme that Revus will work on. If it's not mutated, the enzyme created will be susceptible to Revus, which is good. If you have a hypothetical strain, where you have a mutated version, which would create an enzyme that Rivus can't work on, and a non-mutated, because this trait of resistance is a recessive one, it means so long as you've got a dominant non-mutated gene, the enzyme again would be susceptible to Rivus. What we have with the 43 strain that is resistant to Rivus, and obviously OSBPI chemistry as well, is we have a double mutated gene that CAA chemistry can't work on. So that is what we've selected for in Europe, and hopefully we can keep it at bay in the UK. How the 46 strain differs is with the fact that it is a triploid. It has 3 chromosomes rather than 2. And so long as one of them chromosome arms is carrying that non-mutated gene, then the enzyme created Rivus will work on. Does that make sense? Okay. Now, to make things slightly less clear, you know, there's always a little bit of muddiness with these things. Europeans have created more issues in the sense that they selected for a version of 46, which is a diploid, and it's dropped this chromosome arm. So now all of a sudden you've got a 100% chromosomes that are carrying that mutated version. So we can still select for a 46 strain that is resistant to CAA chemistry. So we need to maintain these practices that we're doing to make sure we don't allow this strain to get out of hand. Now, I just wanted to highlight, and I'm not saying I know exactly what you're doing when you're genotyping, David, but I wanted to highlight that just knowing the genotype doesn't necessarily mean you are going to know exactly how resistant or susceptible it is going to certain chemistries. You are, David, are looking for 12 satellite simple sequence repeat markers within the DNA, which gives a very good indication on what that genotype is going to be, but it doesn't necessarily tell you the traits of resistance, does it? So it's just seeing, for example, I chose the wrong one. Look at that, it's really bad. Let's have a look at the EU13 strain. This is the one that is phenylamide resistant, and you can just see how much variety there is within that genotype. So knowing the genotype is not necessarily the full story. So we need to be careful about that, and we need to obviously be thinking about what David's talking about, of understanding what mutation is there and expressing that to fully understand what trait of resistance these genotypes are bringing. So I'm getting to the point. This is where we're combating that resistance. So a lot of the trials work that we do both in the UK but also across Europe is very important to demonstrate the theory around controlling these resistant strains. And I think there's no better trial that I picked out of the data set that I look at, where there was a Nordic colleague of mine looking at compromised chemistry in the form of mandipropamid in the presence of EU43 and how we can utilize solo chemistry in the best possible way, both in a mixture with, as in this case, fluazinam, but also then a strategy approach of using all the actives that's in our toolbox, which is from the Nordics, this is what they play with. And what the data was showing was, yes, you will inevitably get a level of control by solo actives, but you will then also select for their problems as well. And you can see that mandipropamid and also fluazinam are delivering a level of control, but not great. Just by mixing them together, you are reducing that population of blight down to a bare minimum. And then the strategy approach of mixing and alternating, you are literally down to no blight at all. That is the right approach, and that is the approach to carry on with doing exactly what you are doing. These pictures kind of demonstrate that really quite nicely. But also, it's a level of mutation that we find within these plots. You can see that the untreated obviously is not carrying any mutation in there, but nor is there any crop. By solely using mandipropamid, you are selecting for that resistance, and inevitably you get poor level of control. Fluazinam doesn't select for that mutation, but also is producing no crop at all as well. The best way of doing it is to use them actives in combination with each other. You get a crop at the end of it, and then you are deselecting for resistance. I just wanted to highlight the amount of actives we have, very similar to that table at the start, but also highlighting the restrictions that we have with these actives that do inevitably limit how many times you can apply them, and also understanding the benefits that they're going to bring within a blight program. What I've tried to do here is break that down to the amount of times you can apply each active, and what we want to do is mix these and alternate them through the program. And I've devised a 16 spray program that is strong and robust, using the contacts and the systemics and the actives that have zoospore activity at the right time to ensure that we can both keep a crop there that gives you the produce that you want, but also we're limiting the ability of this blight strain to evolve. And this is my final slide. I just wanted to highlight within that 16 spray program, I'm using no more than 21% of a single active. This is the way that we need to keep on going to make sure that we keep our crops clean and also not selecting for resistance. That's me done. Thank you.
Okay,
Still on?
so thank you. Moving on to Jan. Jan's a plant geneticist from Teagasc in Southern Ireland, leading the program for marker-assisted selection of breeding, and many of you will know Buster, which has 6 PCN resistance genes, and Dan and colleagues were involved in the early work for producing that. He's run and is leading the European project, a Horizon project called IPMorama, and he's going to tell us something about that.
Yeah, thank you, Ian. And I might have slipped the Buster slide in there, even though it's PCN resistance, so apologies. So we've been talking a lot about integrated pest management, and here's my kind of standard infographic that I use when I'm talking about that to most people. And it's got all of the kind of components that we've been talking about today, the forecasting, cultural controls, a lot about population monitoring, which we've got an excellent facility for in late blight. But the bit I'm most tied to and interested in is these blight-resistant varieties of someone who is partly responsible for trying to create these varieties. I'm very, very invested in not only creating them, but seeing that they're used. And actually, up to now, it's been a kind of disappointing deployment. We haven't seen much of those. There's a whole bunch of reasons for that, and I might mention a couple of them. So I thought it might be an idea, when I say integrated, what I mean for varietal resistance in integrated pest or crop management. And the first thing I'm interested in is that the resistance really positively impacts the management practice. What do I mean by that? Well, you don't put a resistant variety in there for a just-in-case scenario. So if things do go wrong, then the variety is resistant. That's actually a reasonable sounding approach, but it's not taking the best advantage of the resistance. I'm hoping I'll show you that in the next couple of minutes. So what we really want is the resistant variety to be an active component of the crop management. The idea being you'd like to spray less. That's one of the goals of this from our point of view, to reach some of the sustainability goals that David mentioned earlier. And really what we want to do here is to start being intelligent about how we deploy this, and we're looking at some of the specific characteristics of the resistance that we have in that particular variety, and I'll talk a little bit about that in a minute, but partly it's to do with how many of these resistance genes are present. And the most important thing from my point of view, especially in the context of what Andy's just been talking about, is this concept of cross-protection. We know chemicals can break down, we know varietal resistance can break down, but what we know, and we don't always understand exactly how this works, is there's an element of cross-protection where if you use them at the same time in an intelligent way, one prevents the other one from breaking down. Not completely, but it can certainly slow that down, and that's the third and final kind of objective we've got in deploying variety resistance in an integrated way. So, as Ian kindly said, I lead this project called IPMorama. I'll throw the website up at the end so you can go and peruse it at your leisure. But it's basically got 3 goals. It's a bit more than about potatoes. It's got a bit of stuff on wheat and legumes in it, but we won't talk about those not interesting crops today. I get to talk about my favourite one. So some of that is about developing the genetic markers and germplasm for resistance breeding. I'll give the example of Buster in a second, just really, really quickly to show what I mean by that. We're really trying to produce real-world IPM approaches that integrate resistant varieties intelligently, and that's what the main interesting part of the project is. And I'm going to show you some of the earliest trials we've done that to test some of the hypotheses we have and how that's worked out this year. And as well as that, what we'll do is an iterative approach. So we're starting to take what we know now and apply that knowledge in trials to test those approaches now. But we're also trying to develop tools and resources to improve the decision support systems that support the intelligent use of varieties in integrated pest management through the project. And what we'll do is every year we'll improve our trialing process. And at the end, we have actually got some farmer scale demonstration trials with our best practice to see whether it works and whether it can live out there in the real world. So I thought it might be useful to talk about what the basis of blight resistance in varieties actually is. Blight in varieties, by and large, it comes from these individual genes, resistance or R genes. They're derived from potato wild relatives, which have co-evolved with late blight. If you look at the current crop of, say, newer varieties that have resistance coming through the pipeline at the moment, most of that resistance is based on about 7, possibly 8 genes of the R8 and R9, which are the remnants of the genes that were introduced in the 60s and 70s by Black, and then some more genes from other wild species that have been introduced in the meantime. How resistance genes work? Well, they're basically they produce a protein which is a kind of surveillance or receptor molecule. So when blight attacks a cell, a plant, it's injecting these effector molecules into the cell, and the point of these molecules is to take over the potato plant as part of the invasion process. These receptor molecules are basically a glorified switch, so when they detect an effector, something that happens, and what happens is it switches on an immune-like response in the cell, and it's a process called the hypersensitive response, in which the invading cell is then killed off. It commits cell suicide, so the blight can't get any further into the cell, and that's what it. This isn't a very good. This isn't actually blight attacking. This effector has been injected directly into the leaf, and you can see here. This effector is producing an immune response, but the important thing is that we have what's called strain specificity. So like Andy demonstrated a minute ago, where you get small point mutations in the genes that are the targets for chemicals, effectors can also mutate, and then, of course, suddenly they don't get picked up by the receptor, and they can evade reception. And then, although that looked clean, what's happened here is it hasn't been, it's the start of the infection process, and this strain can now go on to infect the blight, the plant. Okay, so earlier I said it's been a bit disappointing about how much varietal resistance has been used to date, and you could argue, say maybe 5 years ago, people would have said, well, Dan, the reason for that is there's not a lot of good blight-resistant varieties out there. And I'd argue that situation has pretty much changed now, so I didn't spend a lot of time here, and these are not all of the possible blight-resistant varieties, but I went through and tried to gather a few of them, and these are all things that are based on these resistance genes that I talked to you about a minute ago. Rather than talk about what wild species they are, it's kind of important to keep track of what gene is in what variety. So the Dutch growing organisation, B.L. Akkerbouw, has actually come up with the Dutch breeders with this group system in which they've actually grouped varieties according to which gene they've got, but they've given it either a colour or a letter name. So now we think of these as Group A varieties, they all have the same gene. Group B varieties, they all have the same gene. The point really is that, in actual fact, most of the resistant varieties that we have at the moment each only have one of these R genes, and that's a little problematic. I'm going to show you why in a second. So you can see here some of the R genes. These aren't real varieties. These are Desiree, which has been transformed, it's been genetically engineered to have some of these resistance genes. And what you can see here is one of the things we observe with these R genes is they're not all completely effective even within the season. So they have a certain capacity to delay infection. So this is RPI SHAC1, and you can see here it's got a reasonable delay, and you're running up through the season to STO1 and PTA1, which have an excellent delay. And the important thing is that when you start putting these things in together, we call this stacking, then you get a much, much stronger and more durable effect. So these are stacks of 2 and 3 individuals, and that's where we want to go with conventional breeding and varieties. And the important thing is that because the current crop of varieties are all these single genes, this is like the single site chemistry example that Andy talked about for resistance genes. And so we've got to manage them very carefully while we transition from that point into these dual and multi-resistance stacks. And that's really, really important. Don't repeat the mistakes of the past, as a certain Prime Minister said. So a little bit of shameless self-promotion. I didn't have a really good example of the capacity of breeding to stack blight resistance genes because we're just in the process of doing that now, but we have used a process called marker-assisted selection, which is kind of using genetic fingerprinting to track genes in throughout the breeding program and shepherd them all into the same clone. This is especially useful if those genes have overlapping specificities, so you can't always tell if both of the genes are in there at the same time. And that's how we produce Buster, which has these 6 PCN resistance genes, which makes it incredibly resistant to both species of PCN. But the point here is we can do the same thing with blight, with the genes that we've got, and more genes that we've done coming through the pipeline in terms of gene discovery programs. So while we've got conventionally bred dual resistant varieties coming onto the market now, immediately following those, there are what we call bigger stacks, which we hope will be even more robust. So what would a variety-centric IPM strategy look like? So what I want to show you very briefly and over a couple of slides is, this is hot off the press. We had some trials in IPMorama, which we call the small-scale plot trials. And the idea there is to figure out what we would think, how we think you would operate this now. And then, as I said, over the years, what we'll do is improve that with the results of the of some of the other aspects of the project. And so what we did was to take the susceptible variety Fontan and then just varieties representing the different resistance groups and have them in a multi-location trial in Invergary, where David is, in France and in Poland. It looked at, this is the Polish trial, they all looked a little different, but they had the same basic design. And the idea was that we would compare common practice, and that was just whatever the farm manager normally used, using sort of weather-based prediction to manage that. And all varieties in those blocks would be managed the same way, so that's the blue blocks here. And then what we call a variety-centric IPM approach, and the primary strategy here is to delay the first spray as long as possible, relying on the resistance. And then here's the difficult bit. Ideally, just before the resistance breaks, start spraying and going in full things. That's the magic trick we're trying to pull off, which I think is going to be the most difficult thing to do. And in this case, rather than managing everything at the same time, each resistance group, each little block is treated differently on the basis of that kind of prediction that I showed you earlier about how strong the resistance gene is, so to speak. So, okay, so I've got 10 slides. If I go, it might take 2. Just very clearly, and please don't ask me questions because I don't know anything about decision support software, but this is the decision support that was used. The only thing I want to show you here is that it takes into account, it's from the Farm Maps group of programs. There's an application for late blight in there, and in that you can actually switch on a function which says, I know what resistance is in this, and it starts to use that within the decision support. As I say, it's based on those kind of things I showed you earlier. And then it might give you one set of advice here for Fontan, which it said spray here, but it didn't say spray here for the resistant variety. So very, very quickly, what the results were for that. The trial sites, we had 3 of them. As it happens, there was low pressure in the UK and in Invergary. This is a site in Poland, which I'm not going to try and pronounce. That was kind of a normal-ish pressure site. And the site in France, in Brittany, was very high pressure. I've got to remember to say to you that, in actual fact, these were both full season trials extended to about 90 to 100 days. For some reason in France, they burned off after 60 days. So this got foreshortened a little bit. So you can see in common practice, there were 12, 13, and 9 sprays in this shortened season. If we look at the IPM delimited system within Fontan, because it's susceptible, similar numbers of sprays. But now we're using the IPM using the groups. So they weren't all present at all sites. You can see that in Invergary, we got to the end of the season without spraying again, supported by the DSS. there was no need to spray it. So we went from having 13 sprays to 0. The medium pressure one, well, that went down to 1 spray for these. So there was an interesting breakdown in one of the resistance groups. If it breaks down this year, it may not break down next year, but that's an interesting example of the fact that resistance can break down. And in the high pressure, we didn't have very many of these here, but it basically halved the number of sprays we did. This was described to me as a war zone for blight. It was really put in a very, very high pressure site. So that's where we were. That's really just hot off the press results that came out last month. What can we do to augment that to increase the number of factors we're using in our decision support process for future iterations of those trials? Well, what we'd really like to do is have some sort of in-season early warning network to not just spot the distribution of blight around the place, but how it's when our genes are breaking down, preferably in real time. So not everything that comes out of Europe is bad, Andy. Here's a nice. They've started to do this in the Blight Tracker app in Euroblight, but I wanted to show you this one, which is an application that's been developed by Geert Kessel at Wageningen University, and it's actually one of its features is you can filter notifications from scouts by resistance group, and so it gives you, if you've got that resistant variety and you see that breaking down near you, we hope to be able to start to include that into the DSS, and that's what gives you the early warning of spraying just before your thing breaks down. It turns out that the pattern of strength didn't really work very well because that changes from year to year depending on what population's available. And Andy did me a really big favor in explaining the idea that the groups system using the SSRs, we seem to be dumping on you a lot here, Dave, but it's just to get to the point, it's super useful. It's very, very good information to have this. But yes, it doesn't tell you anything about within a group, it doesn't tell you anything about the likelihood of it being resistance breaking, and that goes also for the resistance groups in varieties as well. And actually, at the James Hutton, and this is Dave's group specifically, we'll be developing rapid genotyping approaches within those to start to track what the resistance breaking capacity of these, and so we'll have an extra layer of predictive stuff that again we can feed into DSS. And so there we go, I'm finished, thanks to all these people who did all the real work and sent me here just to talk about it. Have a look at our IPMorama website. It's really simple. It's ipmorama.eu, and these are the people involved in that. Thanks a million.
Thanks, Dan, and thanks all. We're already over the time allotted to us, so we're going to have to stop. And if there are questions, these guys are going to be around all day. Graham, is it fair to say we need to stop? Well, I want to show you, before we have questions, I want to show you one thing. No. I'm going to jump straight to this one. And this is, I just want to remind people that GB Potatoes are holding a survey and they want everybody to take part. It's an industry confidence survey looking at grower sentiment, production trends, and factors shaping the future of the industry. And they're hoping that this will come out in an annual confidence and production report. It will only work if everybody takes part in it. And it's not a very long survey, and you have until the 16th of January to take part. So just a request to please do take part in it.